Bismillah, Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity. My name is Sari Haryanti, researcher in Medicinal Plant and Traditional Medicine Research and Development Center, or Balai Besar Litbang Tanaman Obat dan Obat Tradisional Tawangmangu. It's an honor to present our research today with the title Ficus septica, an ecosystem keystone species, induced mediated cytotoxicity in Hep-G2 hepatocarcinoma cells. Ficus septica is belonging to Moraceae family and commonly found in various types of ecosystem. It is one of a keystone species, serving as a food source in all seasons for some animals. The presence and role of keystone species have an important impact on other organisms in the system. Ficus septica has been used in traditional medicine for a long time. The leaves are used to treat colds, cough, fevers, diarrhea, rheumatism, and headache. Several research reported the ability of the leaves to inhibit the growth of breast cancer, fungi, and bacteria. Meanwhile, hepatocellular carcinoma is the most common type of liver cancer, accounting for 90% of incidence. The most prevalent cytotoxic medication is doxorubicin. Despite the fact that doxorubicin has a wide range of anti-tumor action, systemic dosing can often result in fatal cardiotoxicity and other negative side effects. Therefore, alternative sources of the discovery for novel anti-cancer agents are still urgently required. The potential effect of Ficus septica for hepatocarcinoma treatment have not been explored widely. So, this study was purposed to investigate the activities of Ficus septica leaf and doxorubicin on Hep-G2 hepatocellular carcinoma cells and varicose line as normal cell representation. We determine the cytotoxicity and evaluate the mechanism through cell cycle profile and intracellular reactive oxygen species. We carried out this research by collecting Ficus septica leaves and then dried, grinded, and macerate with ethanol 96%. The macerate were dried in the oven 50 degrees Celsius to obtain ethanolic extract. Cytotoxic activity was determined by MTTSA. Cells in 96 well plates were treated with the series concentration of extract or doxorubicin for 24 hours. Flow cytometry was used with tropidium iodide staining to evaluate cell cycle profile and DCFDA staining to identify intracellular ROS. The research analysis used linear regression to show correlation between concentration and cell viability to attain IC50 and selectivity index. Flow cytometer data was analyzed using BD Accurate C6 software. This is the pictures of Vero and Hep-G2 cells with 200 magnification. Cells treated with the extract or doxorubicin for 24 hours reveal morphological features such as shrinkage and rounded, resulting detachment from culture disc. This might be the indication of apoptosis. The yellow arrow pointed cell which undergoing cell damage and death. Apoptosis is a kind of programmed cell death with a highly selective process. Cancer cells have an ability to avoid the apoptosis process. So, apoptosis induction plays an important role in the treatment of cancer as it is a popular target of many treatment strategies. The MTTSA was applied to determine cell viability. As you can see in this graph, the extract suppressed Hep-G2 and Vero cells growth with the IC50 around 50 microgram per milli for Hep-G2 and 286 microgram per milli for Vero cells with selectivity index value of 5.6. Doxorubicin had more potent activity with the IC50 0.8 for Hep-G2 and 12.8 microgram per milli for Vero cells with the selectivity index of 16. Several references mention that the selectivity index value greater than 3 are indicated to have a high selectivity towards cancer cells. Next, we determine the impact of the extract to Hep-G2 cell cycle progression using flow cytometry by propidium iodide staining. These graphs show that the extract 10 and 50 microgram per milliliters induced G0, G1 cell cycle arrest with proportional decrease in the S phase. Doxorubicin induced cell cycle arrest at the S phase with proportional reduction in G0, G1 and G2M phase. Cell cycle arrest is one of the cancer response to repair the damaged cells. The failure process will lead to the cell death. We have determined the quantitative ROS production in Hep-G2 and Vero cells by flow cytometry with DGFDA staining. The extract increased intracellular ROS in Hep-G2 significantly by 1.6 to 2-fold. Interestingly, it lowered intracellular ROS levels in Vero cells by up to 10-fold. This finding supports the selectivity of the extract toward hepatocarcinoma cells. Doxorubicin also elevated ROS in Hep-G2 cell while having slight impact on Vero cells. Excessive ROS production can induce oxidative stress leading to the emergence of various diseases. ROS plays dual role in cancer. First, ROS facilitates cancer cell proliferation, survival, and adaptation to hypoxia by promoting pro-tumorigenic signaling. Second, ROS can activate anti-tumorigenic signaling and cause cancer cell death due to oxidative stress. Therefore, increasing oxidative stress in cancer cells has been considered as a promising therapeutic approach for cancer therapy. So, from all of the obtaining data, we conclude that cytotoxic activity of Ficus septica leaves ethanolic extract in Hep-G2 cells are associated with excessive ROS production, leading to oxidative stress and cell cycle arrest. The extract shows high selectivity index to Hep-G2 hepatocarcinoma cells compared to normal Vero cells. Further research are required to isolate active compound and conceive specific mechanism involved in Ficus septica-induced cell death. That's all my presentation. Thank you for your attention. Salam sehat, sehat Indonesiaku. Wassalamualaikum warahmatullahi wabarakatuh.